Okay, each person today should have received a packet. The first 2 pages of the packets are your notes. This last page is going to be for you to turn in. Please listen to these directions. You do not all have the same worksheet. So you cannot copy me. You need to listen to the directions and follow the directions on your own sheet. The first thing we need to do is to come over to the identification key. And we need to label what everything stands for. So the P stands for phosphate. The backbone of every molecule is made up of phosphate and a sugar, and that sugar is deoxyribose. So our backbones are made up of phosphate, and deoxyribose sugars. Now we need to label what all of these bases stand for. So the A stands for adenine. The C stands for cytosine. The T stands for thymine. And the G stands for guanine. And we remember with our base pairing rules that straight letters go with straight letters, or apples are in trees. Curvy letters go with curvy letters, or cars are in garages. So what we're going to do is we're going to make a model of DNA and then we're going to replicate it. So now that we've labeled everything, we need to look at our parent strand of DNA. And we need to use the base pairing rule and fill in the missing base pairs in the parent DNA molecule. Everybody has a different parent DNA molecule. So fill in the matching bases for your molecule. On mine, cars go in garages. Garages have cars. Adenine goes to thymine. Thymine goes to adenine. Garages have cars. Adenine goes to thymine. Next, you need to color the base pairs in the following colors. On your sheet, every A you have on your sheet will be green. So I'm going to color all my adenines green. Every thymine will be yellow. Your thymines should be next to your adenines. I'm going to color every cytosine purple. And I'm going to color every guanine red. Your guanines should be next to your cytosines. So purple and red will be next to each other, and green and yellow will be next to each other if you did this right. It says color the backbone of the parent molecule light blue. So you're going to color this backbone and this backbone light blue. Now we need to replicate our DNA or make a copy of it. So when DNA makes a copy of itself, the parent DNA molecule splits down the center and separates. So this half of the DNA molecule is going to come down here. So I am going to put a piece of paper over so I can't see the other half. And I'm going to copy this line, whatever this line is on my sheet, on the left side down here. So on my sheet, I have C, G, T, T, C, A. Now I'm going to make the other half of my DNA. Remember, replication is semi-conservative, so we're going to split the original. So now I'm going to use the right half, and I'm going to copy it down here, whatever your right half says. So mine says G, C, A, G, T. And then we're going to color those original parent molecules just like we did at the top. So we're going to color all of our A's green. We're going to color all of our T's yellow, all of our C's purple, and all of our G's red. We're going to color the backbones blue because those are still our original molecule. Now the DNA is going to replicate. So we're going to match again. For every C, we're going to put a G. For every G, C, thymine has adenine. Thymine has adenine. Cars and garages, apples and trees. Garages have cars. Cars have garages. Apples are in trees. Apples are in trees. Garages are in cars. Trees have apples. And then we're going to color them to match. So we're going to color every adenine green. We're going to color every thymine yellow. And if you did it right, green and yellow will be together. We're going to color every cytosine purple. We're going to color every guanine red. Now, these are new strands of DNA. It's semi-conservative. It has one strand of old DNA and one strand of new DNA. So on our new DNA, we're going to color those backbones orange. When you're done with this, you need to make sure you answer the questions at the bottom. There's 2 questions at the bottom to answer. And then you need to come up here and write a summary of DNA replication. It can be broken down into 3 steps. So in this box, you're going to list the steps 1, 2, and 3. You can use your notes for this. Your notes have it laid out for you. When you are done with this, rip this page off of the back and turn it into the box. Make sure you have your name on it. And that is it for today.